Good evening, friends. Welcome to this month's monthly lecture of the Varahamihira Science Forum. I'll give a brief introduction of the forum and the speaker, and then we'll turn it over to the speaker of the day. Varahamihira Science Forum was started about 5 years back. We are on several social media. We are on Facebook, we are on Twitter, Instagram, and YouTube. We also have an active WhatsApp group. So three of the organizers' names, myself, Mohan and Ramki, our names are there, our numbers are there. You can just message us and we'll add you to the WhatsApp group if you want. You can always go to our Facebook page, Twitter, Instagram, just search for Varahamihira Science Forum and you'll find it. Videos of previous events are all there. Notifications of all future events are also sent via social media. In the WhatsApp group, we try to occasionally have active discussions on the lectures of the day. We started this a few years back. At that time, people were talking about cultural capital and all that. UNESCO had recognized Madras. The city of Madras is actually famous for Carnatic music concerts in December, and of course the film industry. But we wanted to get the public to feel that science also is part of our culture. We've had a few scientific lectures in the city, but they are generally very technical. They are not necessarily for the general public. So we wanted a forum where speakers with either experts or even amateurs with a lot of knowledge could deliver popular lectures to target not just as the professionals in the field, but at the public, the curious, scientifically curious public. So we thought we'd do it outside, the standard places like the universities and colleges and hospitals and conference halls. And so we were not sure how long it would succeed, but we are now in our 5th year. So we are very delighted that people are still listening. Of course, the last 2 years since the Corona lockdown in March, we've gone online. And the advantage of going online is that we don't need a speaker's physical presence in the city. Before that, for the 3 years, we've had lectures at Tamil Virtual Academy in Chennai. But of late, we've been the last 2 years we've been having these lectures online. So we've had the pleasure of inviting speakers from different parts of India and even different parts of the world. We've had 3 or 4 speakers speak from the United States and England and other places. And so we hope to continue to do this in the near future. One of these days, we'll probably come back offline. When we do, we'll notify all our regular subscribers on all the social media. So this is just a sample of some of the viewers we've had, or some of the lecturers we've had on our program. Arvind Gupta, former professor of IIT Kanpur, did a very popular lecture titled Science Through Toys. K.V. Balasubramaniam, a meteorologist at the Chennai Meteorological Center, did an excellent lecture called Weather and Climate in Sangam Poetry. So he took the most ancient Tamil poetry and added references to it about weather and climate in those Sangam poetry, and gave it context in terms of current meteorological understanding. We had Dr. Uttara Dorirajan, who's a professor of physics at D.G. Vaishnav College, give us a lecture on Leelavati's daughters, the less well-known women scientists of India. Recently, we had Aparajit Ramnath, a professor at Ahmedabad University. He gave a lecture on the birth of India's aircraft manufacture industry, which is kind of a very recent kind of phenomenon. And coming to today's topic, mostly about, primarily about medicine, we've had a few. So we had topics, of course, lectures of course, very different topics: physics, chemistry, technology, history, and so on. But for today's lecture, I'll just give a sample of the medical lectures on medicine that we've had. One of the earliest lectures, I think this is the very third lecture that we had, is a lecture by K. Sivaraman on the hidden phages of medical history. This is about bacteriophages. He gave this lecture in October 2017. Then we had Dr. Relenu Ramachandran, the world famous neurologist, who's a very good friend of the forum. He gave a talk about relevance of Freud in modern neuroscience. This is our case center. And we had a youngster, a very young person, Nishant Chandrasekhar, who just freshly graduated, did his PhD. He gave a talk, an introductory talk on neuroscience. And just after the virus, the corona, the lockdown started, we had Ramanan Jagannathan, one of our organizers, give a lecture on CRISPR, gene editing tool, and he also gave a talk about viruses and information. Viruses is the spread of viruses. We had a lecture by Dr. Ramanohar, who is an Ayurvedic doctor at the Amrita School of Ayurveda. He gave a lecture on is Ayurveda relevant? And that brings us to today's lecture, which is by Dr. Nimesh Gupta, scientist and head at the Vaccine Immunology Laboratory at the National Institute of Immunology. I'll give a brief introduction to the speaker. Dr. Gupta is heading the Vaccine Immunology Laboratory at the National Institute of Immunology at New Delhi. He's a virologist and an immunologist by training. He was trained for virology and immunology at the Pasteur Institute and the Pierre Curie University in Paris, France. He now leads a team interested in understanding the immunological basis of long-lasting protective immunity, and this is very relevant to our times today and has come front and center. The long-term goal is to identify attributes of CD4 cells, T helper cells, etc. The human T cell immunology program is, that's his major field of research. Several national and international funding agencies are in charge of funding that. He is received global recognition as the Martin Villar Fellow for his approach in this particular subject. He has received several awards from prominent international societies for his contribution to the field of T cell immunology and the advancement of vaccinology. Some institutes like European Federation of Immunological Societies, the American Association of Immunologists, and the Merieux Foundation. He's contributed several publications in prominent journals in the field of virology and immunology. He has national and international patents on the applications of T cells and immunotherapy. In COVID-19 research, his team is working extensively to determine the quality and durability of immunological memory, with emphasis on T cells in SARS-CoV-2 infections and vaccination. This is so relevant. So I'm very, very happy that he agreed to give this lecture for us. And sir, without much further ado, I'd like to invite you to deliver your address for our audience.
Yeah, thanks so much, Badri, and thanks so much, Goku. I think this is something which is, I must, let me first share the screen, and yeah. Hope you can see the screen and everything is fine.
Yes, fine.
Perfect. So let me, again, thank the organizers from this forum. This actually comes under a Science Setu program of our institute, National Institute of Immunology, and I have been almost vigorously engaged in this activity of spreading the awareness on vaccines, and particularly with this pandemic. In past 1 year, I have been going to multiple places virtually, in the colleges, in the schools, in the clinical centers, and to say that what we can understand, what we can know, what we should know about the vaccines, because with COVID-19, I think it becomes a bit easier job for us to slowly get more awareness, slowly get the society much more aware about the vaccines and how do they actually work, why do we need them, and what are the ways which we are actually working to make them much better in terms of their future utilization. So when this, we have a PRO in our institute, Jyoti, when she came to me and asked about the lecture in this forum, I was a bit reluctant initially because I was not much aware of, how such things can be utilized for the awareness. But then I realized that, as we go to institutions and schools and colleges, I think this is also one of the great responsibility that we can take further to spread the words. And I think you guys are doing a fantastic job in terms of spreading the awareness for multiple fields, not only, sticking to one of the areas. And I think that is really necessary in today's time, that at least we can take the research to the society, to the people. I think that's what was missing in the last decade. So with this advent of a lot of social media, virtual platforms, it becomes easier for everyone to get into these areas. Fine. So thanks to the forum, thanks to the Science Setu committee of our institute to bring me here. We will just talk about a bit on the immunology of vaccines today. Let me just. Okay. So before we, get into it, as the tradition of the Science Setu program is, I will just give a brief introduction about the institute where I am working and where this program is actually, initiated in the country. So National Institute of Immunology was set up, sometime in 1981 as an autonomous research institute under the Department of Science and Technology. And then, it was transferred to the Department of Biotechnology in '86. And we can say that DVT started from the inception of NII, and from where it then started with with the multiple autonomous institutes in the Department of Biotechnology. Still, it is registered as a society, and there is a mission of this institute to actually undertake and promote a lot of the research which is coordinating around the basic and the applied immunology. And it has a lot of other areas which are actually linking very effectively with the immunology and to promote and to enhance the expansion of this particular field into the different areas, not only sticking to vaccine development, but also in the infectious diseases, and now slowly from autoimmunity to cancer. It's not only the research center. We do have the PhD enrollments, and we actually generate and develop a lot of manpower in these areas, which the research is going on here. And along with that, we have other courses, workshops, symposia, and training programs, which actually nurture our, the young Indian population in these fields of immunology, vaccine areas, vaccine development, and the other related areas. So, of course, the goals of this institute are to provide national and international leadership in this immunological sciences, to provide with the quality solutions to the healthcare, and also to expand, some of the tools that we can generate from biology, chemistry, physics, or mathematics to then incorporate into the immunological science to see how we can really take it further to the next level. And we have some of the real outcome from this institute in past decades in terms of the vaccines, like the anti-leprosy vaccine, which is already has been used in multiple diseases now, as it has been shown with the off-target effects. We are also, capable of taking out some of the cancer immunotherapeutics on the basis of dendritic cell-based vaccines. And also there are, the laboratories which are working on a lot on the product development cell, particularly in the area of autoimmunity. So currently we, are majorly emphasizing on understanding the human immune system, how vaccines work, and how we can really design the newer, or let's say much more potent vaccines for the infectious diseases. And we are actually now at the interface of a lot of basic immunology as well as the translational immunology in the domain of infectious diseases. And of course, we provide a lot of good information, foundational information in the field of immunology from our institute in terms of like publications or the patents. Fine. So with this introduction to the institute, now what we will do today, as initially mentioned by Mr. Gopu, that it's like a lot of non-technical, information and non-technical lectures are required so that we can reach to the society. And even if we can give them a one understanding, I think that should be a success for that particular lecture. So non-technically, what will I do here? I will try to cover up, the domain of vaccinology, starting from what is a vaccine, a very brief information. We will get into the history, because I think the history of vaccine is something which is so enriched and it's so important that tells us and that also tells the youngsters that how the field is evolving, how the field evolved, and also how we can actually take it further. What are the ways where we have reached until here? It's almost like more than 200 years of the vaccinology, which we are at this stage. And a lot of these new vaccines which are coming up has, a lot of information, a lot of knowledge from these historical developments. We will then get into the details, which is, I will not say a very old or a very historical area. How vaccines work is something which is now slowly coming up with the advent of understanding the human immune system. So initially, we were not much aware of our own immune system. The past decade was really tremendously helpful to understand the human immune system, how the immune system works. And as we started learning that how the immune system works, now we can actually ask the questions that how the vaccines are working, right? We have so many vaccines in our national immunization programs. We know they confer protection, but we do not know how do they confer protection. And that becomes very important if we know that how do they work, so that you can then rationally design the new vaccines. And that's the very key of our laboratory, my team here at NII, to understand how vaccines work in humans, so that we can develop new rationals for much more potent vaccines. And still we are in the pandemic. We are not yet over from this pandemic. So I will touch upon the kind of COVID-19 vaccines we have, and what is the immunology of those COVID vaccines very generally. But I will also provide some information on the work which is going on in our laboratory in a very superficial manner, that how we are actually helping or contributing in the country in terms of understanding the immunology of the COVID-19 vaccines, and we end up with some of the takeaways. So let's start here. I think in a very general scenario, what we have to understand that what is a protective immunity, right? That is the basis of particularly the vaccines. So what happens in our lifetime? We are exposed to a plethora of pathogens, so many pathogens that we see in our lifetime, starting from birth to adult to the senior level. And we do not develop disease because of all these pathogens that we are exposed to, right? So there are two different mechanisms exist here. One of them, which was well known, was once you develop an infection, once you develop a disease, you then develop an immunological memory against that particular pathogen. That may last lifelong, but that is not necessarily lifelong in cases of all the pathogens. So that was one of the well-known, phenomena that we started learning when we were actually learning about the immunology and the protection, via our exposure to the pathogens. The second possibility that we did not realize before, but now we are started appreciating it on the way as we learn more about the different pathogens, is a scenario where you develop the immunological memory, but you do not develop the disease. And this is what we call the asymptomatic infection, where you do not show a clinical manifestations, but you do expose and your immune system do fight against that particular pathogen, and there is a memory formed. So these are the two ways when we actually develop immunological memory, which we can say as a protective immunity that we have developed against X pathogen via virtue of either developing clinical manifestations or other way around without having the clinical manifestations. Now, this is something which was the basis for coming to the development of vaccines. Vaccines do actually provide the development of memory. And that's where we come in between, where the vaccines do not supposed to provide us infection or disease, but provide the immunological memory without creating what a live pathogen can actually do to our body. So the protection conferred by these different ways may be different. Sometimes the mechanism may also be different, but the output is that you actually develop some memory, so that in future, when you see that particular pathogen, you come back and you fight against that pathogen, because your body have already seen that particular pathogen in a kind of little compromised form, we'll say attenuated, inactivated subunits, something that we come upon in the consecutive slides. So I think this is something which is very important, has been said by Stanley multiple times, and I think it becomes a notion in the field that the vaccination has helped our population, has helped the human, enormously. So the public health, particularly supported by vaccination, I think there is no comparison. And we say that, okay, except the safe water, there is no other modality that actually can help us in majorly reducing the mortality as well as giving the population growth. and actually enhancing the life expectancy. So this is something which is very important to understand, that vaccines are one of the most important pillar when we talk about a better life expectancy in terms of a particular society. And this is an example, right? We talk about theoretical scenario, a hypothetical scenario, and smallpox came as an example that if you use vaccine, yes, you can actually eradicate a disease, right? So we always use this as an example where it's a successful example of a vaccine, which says after 8 years, you did not see the smallpox because your vaccine actually helped to eradicate it. Multiple ways, multiple mechanisms involved, which probably some of them have been discovered, some of them still needs to be explored. So when we talk about this, something which goes back is the particularly the history of the vaccines, that how these things actually started. So what will I do here? I will not get into the details because history of vaccines, particularly as I mentioned, that it's almost 200 years, more than 200 years of accumulation that we have in the domain. This particular duration may not be sufficient to touch upon. So I'll just give you some examples that how it actually goes on step by step while we develop these vaccines. And I think this is something which we all are aware, somewhere in our families, somewhere in our relatives or our surroundings, we have someone, a senior citizen, who actually still had the scars, right? The scars of the smallpox. So the smallpox, this particular image that you are seeing from a kid, it is something from the Afghanistan, and they still found that even during those years of 50 to 70, this disease was very much reported, documented on the planet. And on the right side, if you see, this is something which at that duration, at that time, was a kind of advertisement that was trying to see that whether we really eradicated the smallpox from the country or cleared it from our country or not. And very interestingly, it was asking that, okay, if you want to win rupees 1,000, you just have to inform that you find someone a fresh case of smallpox, and if that's correct, then you will actually get rewarded because we want to clear the smallpox. How it started, how we get the confidence and the gain and with level of the vaccines, which we think that we can help us to clear this particular infection. And that was, I think, a very famous Jenner's experiment that probably many of us are aware, is when he started sometime in 1700. This was a very later stage from 1500 or 1600, when this variolation was started somewhere in China, India, and all other countries, from Turkey and other places. Sometime in 96, close to 1800, what Jenner's found, that a lot of smallpox cases were happening in his surroundings in the village, but particularly the milkmaids were protected. They were not actually getting the infection. And he was a bit curious that why the milkmaids never got this infection. And what he found, that milkmaids actually have something related to smallpox, which is another virus, which is a cowpox. And these individuals with cowpox, they don't get infected with the smallpox. And with cowpox, they just don't have very severe outcome of the infection. It's like something, very few kind of lesions which cleared off, after some time. So what he did, he did some of the, the studies, he developed some of the notions, confidence, built the hypothesis. And he also tried from, a little bit pustule from a milkmaid named Sarah. And he took that, pustule, some extract from it, injected in a boy. He was an 8-year-old boy. And he actually demonstrated later that, that boy never got the infection from the smallpox. And he actually told that, particularly the response of our body to cowpox is actually sufficient to give us protection against smallpox. So this is where it started, and it was so famous in those durations where he was actually helping the society by providing this vaccine, that it termed even the area where he used to keep his vaccine, just to protect it from the high temperature, because these are kind of specific dens they make at the duration, which we still have in India in some of the places, which somewhere it's called the Vaccinia temple, the vaccination temple. And he was here, to help, particularly to provide the vaccine, the free of cost to the individuals in this village or the surroundings. But it was particularly the Jenners who actually invented, tried to put up some of the concepts on, the vaccine development, and how we can really protect, by providing such kind of cross-reactive protections. But actually, it was Louis Pasteur who actually came up with a lot of demonstrations. He came up with a lot of new knowledge, which actually helped to establish the vaccinology domain. So he was the one who actually, learned and gained the knowledge on the way while he was actually studying something else, right? He started his work particularly looking into the red wines, because the wines were, a bit sour, and the winemakers were asking him to see that how he can actually control it, because of course it was a fermentation process which he was the master of. But he slowly expanded his areas, and he get into particular this microbiology domain, which we all say is the father of microbiology. And then he provided this knowledge that while attenuating some of these viruses, when we say attenuation, it is in a layman we can say a weakening of a virus or a pathogen, so that we can actually weaken its virulence, so that he does not cause the level of infection, the level of pathogenicity, or the severity that it is actually can cause when it is a very strong form. So that's where he come up with many of the examples, and he showed the way to the vaccinology domain, that how we can generate the vaccines against multiple pathogens. I will just give you some of those examples, as they also are just not the example of vaccine development, but they also give us an idea that how one has to keep his mind always open to the research. Research should not be biased in terms of looking into a particular line of the question that you have asked. Research should be open in terms of whatever the data, the results, the observation you made, you have to think over it, right? That's what some of these examples I will just share for the youngsters. And the best example here is the Pasteur's cholera experiment. So he was working on the fowl cholera, the chicken cholera, and trying to understand how, because it was actually killing a lot of chickens and affecting the particularly the businessmen there, those who were actually having the farms. And he was trying to understand how this bacteria, which is called the Pasteur multicida, is actually killing these chickens. So this is just on the left side. You see this is his a laboratory pictogram from the École Normale Supérieure in Paris. And so what happens in let's not see on the right side first. Let's just hear this story. It's very, very interesting story. What happens during the summer, that he was on holiday. He was going on holidays, and they were actually preparing this bacterial culture, and they're trying to inject this bacterial culture into the chicken, and then trying to understand what is happening, how are they dying, and how actually they are responding to those cultures. So he was going on holiday, so he asked his assistant, Chamberlain, that you just have the fresh culture lying on the table, inject all these chicken, and once I am back after 3 or 4 weeks, we'll see what happens. What happened? The Louis Pasteur went on a holiday, and next day the Chamberlain also went on holiday. So he actually forgot to inject those chickens, and he just left that culture on the table, and it was not even the caught. So when he came back after 3 to 4 weeks, he realized that, oh, he forgot to inject the chickens. So then he took that culture and he injected. By the time Pasteur came back, he asked that, oh, you injected, but these chickens are not dying. So then he thought, oh, I made a mistake. I just forgot the culture here on the table, and just because the old culture I injected, maybe it's not working and they are not dying. And then he told, okay, I will just probably clear off these chickens, and I will start the experiment again. But then Pasteur told, no, I think that's something is happening. Let's see why these chickens were not dying. And what he did an experiment which we always in the vaccinology domain say a very famous cholera experiment, that he took those chickens, those who were injected with that old outside capped broth, and the normal chickens, and he then gave them the fresh bacteria. What he found, that the chickens, those who received that long time capped broth, they did not develop the disease, or they did not die. But the chickens, those who did not receive it previously, they all died. So then he realized that something has happened by keeping those bacteria outside in a long term, that they probably are not virulent enough to cause the disease. And they are the one probably are generating or training the system, the immune system, in such a way that when these chickens are seeing a new pathogen, a fresh pathogen, they're able to fight back. So that's where this concept of weakening the pathogen or inactivating, attenuating the pathogen started. And then there were many developments had happened on majority of the vaccines which were developed by this particular technique of either live attenuation, weakening them, or inactivating them. And he was also involved in another finding where, utilizing the similar the idea of inactivating, he developed a rabies vaccine which he also tried in one of the boy who was, bitten by a rabid dog. And, he was able to protect it by giving him, a continuous injections, which they developed by drying the spinal from the rabies injected rabbits. So these observations that he made that become, the kind of principle for empirical development of vaccines in the field, and many vaccines developed on those particular concepts. And this particular development of the rabies vaccine, which was very successful at that duration, was actually utilized and becomes the basis. This is the statue that you see in the Institute Pasteur, where I got a opportunity and fortunate to be there to get trained, and where I started then my journey on this vaccinology domain and getting into this area where I'm today. So these were, the example of a pastor which he actually put in to a lot of youngsters at that time, and the scientists, that whatever the observation that we see has some sense. And if the technically, the things has to be sound, then your results should be seen very carefully with an open mind. And that actually can lead to a lot of new discoveries that can help the society. So I think this is, a very long journey. If we get into the details of many other very, kind of intellectuals, those who helped the vaccinology domain to grow from the multiple steps, from the different phases of the vaccinology, it will not be easy today to do it. But just, if you see this particular graph, that is, a very, informative graph that actually says how from that era of 1700, 1800, we reached until here 2020, and what level of development we made in the vaccinology domain. As we already talked about, the live attenuated vaccines were developed, and the vaccination were developed with the killed organisms or the inactivated one. Slowly we developed the capacity to provide and develop these vaccines in the cell culture models, and that's actually helped a lot to produce many of these vaccines. And now we are actually growing further in this area, where we are reaching to a lot of omics, a lot of immunological understanding now to actually develop the vaccines. And this particular decade of what we are, and the coming decade, what we will have is a lot of immunology will be required to actually develop the vaccines, not by the empirical approach alone, but also to having the rational, because now we have a lot of knowledge about how we can actually develop a good vaccine by tweaking it, by making it much more favorable to induce what we want, a long lasting protection. So as we learned about a bit of the history of the vaccine, it's more important to get into that area where we should have some understanding about how these vaccines work, because once we know that, I think we will be much more open for the vaccinations. So vaccines work by two different ways. One we call a specific impact, specific, which we can also say on target, which may be a direct effect. And it could also be an indirect effect. I'll come on to that when we talk about indirect effect as the prevention for the onward transmission, which was much discussed in case of the COVID-19 infection, which was not really relevant. And a second area is on the non-specific, the off-target effects, particularly what kind of off-target effects the vaccine can provide. It's an interesting area, maybe more in infancy and much more unexplored area of the vaccinology. So when we talk about a very specific effect, a vaccine actually induce and actually include multiple components of our immune system that started from the innate responses and then leading to the adaptive responses. So I will not get into the details of what is innate, what is adaptive, but I can just in one word I can say innate, where it's not specific. It will probably be the similar magnitude, intensity to the different levels of pathogens. But when it comes to adaptive, it is specific, and the adaptive immunity, composed of T cells and B cells, which are specific to a pathogen. And we may have thousands, millions of these cells in our body, which have specificity to particular pathogen. And this is something which is helping us to keep the memory, because when we know X pathogen, the cells specific to those X pathogen will expand, will stay in the tissues, and that's where they will come back when this X pathogen is again, coming to us. So how this cycle of vaccine immune response works? Let's go into the kinetics step by step. When we receive a vaccine, what happens? So if you see this particular kinetics, the kinetogram that I say is you receive a vaccine, that vaccine components, the vaccine molecules, let's say you got it here, and then the vaccine components are actually released. Let's say an adjuvant is there, a vaccine, different proteins are there, and they will be taken up by the antigen presenting cells. The antigen presenting cells could be dendritic cells, monocytes, and others, the like macrophages a bit. And these antigen presenting cells will take these different molecules of the vaccine to the lymphoid organs, the small lymphoid organs that we have where we actually got the vaccine, which we call draining lymph nodes. And there are two ways. One, the cells, the antigen presenting cells are taking those vaccine molecules to lymphoid tissue. A second way, the vaccine molecules can also reach to the lymphoid tissue via systemic circulation. And what happens when these molecules reaching to the lymphoid organ via systemic circulation, they actually can induce the activation of B cells. And here, these B cells will actually provide a primary guard of antibodies. Some of the antibodies will come out, and that may not stay long, that may not be of a very high good quality, but they will be sufficient to at least provide a way to reduce or to clear the virus to fight with. And that is the first step of B cell activation happens via the systemic way. Second, these cells, which we are seeing here in pink color, they are taking these vaccine molecules and they go to the lymphoid organs. They present the different parts of this protein from the vaccine to the T cells. These are called T helper cells. Once they present it, the specific T helper cells gets activated, and then what they do, they actually go and talk to the B cells which are specific to these proteins. And these T cells, B cells crosstalk, they interact, and then the outcome of this crosstalk, the outcome of this interaction, which we actually call as a germinal center reaction, that takes place. And this germinal center reaction is the basis for humoral immunity development, to have memory B cells, to have bone marrow plasma cells, those who will produce antibodies, and to also have a good quality, high affinity antibodies, right? So this is particularly a basis of generating the humoral immunity in case of the vaccine response. Third thing, what happens in the lymphoid organs is the same cells, the antigen presenting cells, also present the different antigens of this vaccine molecules to the killer T cells. Killer T cells could be both CD4 as well as CD8, and this whole process actually takes 3 to 7 days in the context of when the antigen is sufficiently available. That may extend from 7 days to 14 days in terms of how long the antigen persists here in the lymphoid organs and how long those germinal centers are still actually going on. It's like a kind of a power generating area where you have the fuel, and until you have the fuel as an antigen, you actually generate these T and B interaction, and you generate the output in terms of memory B cells and antibodies. And what happens after this, the whole germinal center reaction, then you have the outcome of this vaccine induced responses in circulation in blood. Antibodies of different qualities, of different abilities, different cells, the memory B cells, the memory T cells, the CD8 T cells, the killer CD4 T cells. And some of them, when stay in circulation, some of them goes to the different tissues to their location, like in bone marrow, they stay in lymph nodes, and they also go to different tissues where the inflammation has happened. So, and they stay there, like in bone marrow, the cells which are coming from the memory B cells can stay for years. Like, that has been example of yellow fever vaccines or smallpox vaccines, which say 80 to 90 years has been, seen that you will find these cells, and you will also find the antibodies in circulation, which are coming from these bone marrow derived plasma cells. So this is an overall, a kinetics of what happens when you see a vaccine, when you receive a vaccine, until you produce a protective immunity, a kind of correlates, of the protection in terms of both the antibodies as well as the cellular responses. And when we talk about these, the different outcome, the different output, still many of these vaccines, we do not know about what kind of correlates, they have a protection, but there are different parameters, there are different endpoints that has been seen that can help us to understand as a correlate. The antibodies, which could be serum antibodies, they may be neutralizing. Neutralizing means they may be inhibiting the entry of virus inside the cell. That's, let's say, neutralizing. They are non-neutralizing. They are also actually working in terms of their functionality as well as in terms of their ability. So this is something which not only goes by binding the virus, which also goes by the binding of the other portion of the antibody, which we call FC portion, to the different FC receptors on the cells. And they work via both functions. Mucosal antibodies, I think much talked now in case of the COVID-19 vaccines, and it slowly gaining the momentum in terms of the new vaccines coming in. And they are actually helpful in terms of providing the protection in the mucosal areas. When it comes to the cellular responses, you have CD4 T cells that comes with multiple protective mechanisms. One which they come as B cell help. As I told you in the previous slide, that B cell help becomes a very important aspect of generating the immune response, generating the immune memory in terms of the humoral immunity. So these are the functions from the CD4 T cells, then cytokines, the killing capacities, as well as the CD8 T cells. So these are the potential immune mechanisms which our our immune system actually provides and confer when we receive a vaccine. But something which has to be noted down very clearly, that the mechanism of protection which a vaccination can provide, it may not necessarily be the same what we can actually get after recovery from the infection. It may vary. So just to quickly rush on for some of our young fellows to understand that how these kind of correlates actually work. If you see like the antibodies, how the antibodies will work, that you have B cells, they produce antibodies. These antibodies will bind to the virus and they help in clearing this virus. Second, these antibodies will bind via their Fc portion to the antigen presenting cells, let's say macrophages and phagocytic cells, and take up the virus here where these cells kill it. So this is how antibodies help in two different ways via by clearing these viruses, and they actually enhance the virus clearance by these innate mechanisms. The cell killing, how does it happen? Is you have the virus inside the virus infected cells, and what will happen? There will be the signals which will be provided by this cell, and those signals will be recognized, received by the killer T cells, whether they are CD4 or the CD8, and then these cells comes in, they talk to these infected cells, secrete some of the cytokines, which will then create cytotoxicity and kill the virus infected cell. So this is how these direct cell killing will actually work, or via these cells, those who are capable of killing the infected cells, and then they reduce the load of virus. They do not clear the virus completely, but they do not allow the virus to spread further. So that actually helps and provide a window of an opportunity for our immune system to come back and fight against this pathogen. Something which we actually talked about, in the previous slide, that the basis of germinal central reaction, the basis of humoral immunity, the antibodies, memory B cells, immune memory, is actually germinal centers, which are developed because of T cell and the B cell crosstalk, that we have these cells in our immune system. Just a small cartoon here, that in these germinal centers, which we have seen before, these cells, which particularly we call T helper cells, and a specialized subset which we call follicular T helper cells, are the ones those who actually provide help to the B cell, help in terms of multiple signals that then allow a much more intensive crosstalk between these cells in the presence of antigens provided by some of the cells called follicular dendritic cells in the tissues, and that lead to the generation of memory B cells, bone marrow plasma cells, and a kind of very good quality and high affinity antibodies. So this is what exactly is the focus in our laboratory, where we are actually working upon, and we are trying to see in the different vaccination models, like what happens after vaccination, how this immune memory is developing, what kind of T cell responses they are generating, and what kind of crosstalk is happening between the T cell and B cells, so that we can come up with a message that, oh yes, this is particularly a very nice information on the traits of these TFH cells. Let's see if your vaccine can induce this. If your vaccine can induce this, then you may have a very long-lasting humoral immunity. You need not to wait for 10 years to look into your data. You can actually see at a very phase 1 trial stage of your vaccine. So these are the things which we are actually looking at, and we are actually taking the comparisons of these vaccine studies with the infection models, and trying to see in the multiple in infections and vaccination, like Japanese encephalitis, dengue, now COVID-19, to find out how does it work, how does this particular phenomena and the biology of immune memory development takes place, what are the role of these cells there, and can we really take out some knowledge so that we can help the vaccinology domain. Recently, we have, in the same line, we have recently developed a very, interesting assay, which can now help the vaccines for evaluation for their capacity in inducing these cells. the cells, those who can actually provide help to the B cells. If we actually can see that the capacity of X vaccine is there in inducing these these T helper cells, which have the function of helping the B cells, which can have the demonstrated function of helping B cell, yes, this could be utilized as one of the qualitative parameter to say the vaccine is capable of inducing probably a good quality humoral immunity. So this work we recently provided in, and if someone wants to go into the details, you can just go through this DOI, and then you will get the detailed idea about how this assay can be utilized, to understand the fundamentals as well as to evaluate the vaccines for their potential in inducing the humoral immunity. So now let's move towards the indirect effects, and I think indirect effects were much talked in this COVID-19 pandemic, but probably is not in the correct context. They were talked in case of the COVID-19 infection. And what are these indirect effects? I think all of us have heard this word, herd immunity. And what is this herd immunity which comes as an indirect effect of the vaccinations? Let's see this example. So you have these blue circles and you have a red circle, right? The red circle here, let's say, is an infected host. Now, if all these blue, which are not immune, which have not seen the vaccine, which have not seen any kind of exposure and are not having any kind of memory, what will happen? Every person who comes into contact of this red person will develop infection. And this is how the infection will actually spread with some certain probability. Now here, the herd immunity comes in this particular manner. If you see this particular pictogram, what do you see? The same infected person, but the individuals with these gray circles are the ones, those who are immune. Now, what will happen? This probability of the spread will go down because this individual will not be able to give the transmission of infection to these individuals who are already immune. And this is what we call is an indirect effect of vaccination, which helps not only the individual by providing direct effect, but which actually helps the society by actually reducing the chances of spread. And if we come down to the another part, which we have seen the non-specific, part of the vaccines, which we say the off-target effects, these are kind of the different downstream or the lateral effects. I will not get into the details of it, but I can just give you an example that off-target effects are like, if you get a BCG vaccination for a certain duration, you may see that you are also protected against other pathogens. Particularly, many of the studies have been done in the early life, where after the BCG vaccination, the studies found that the children were actually protected against the severity by multiple pathogens if they received it. And there were some very good correlation. Epidemiological data has been provided on RSV, influenza, or HSV2. Although these trials were not designed in such a way that they can actually provide a real answer that why and how it is happening. But I think that becomes very important now as we can see, these pandemics more in future. This particular area needs to be explored because that could be one of the quickest possibility to develop, provide the protections to those who are really working at the front line. So I will not get into the details here, but this is also a kind of the way vaccine actually works, which we call non-specific effects, which we call off-target effects of the vaccination. Now let's get into the next level, the next step after having the understanding on how vaccine works on the COVID-19 vaccines. So I think every one of us are now aware of the virus, which is called SARS coronavirus 2, which actually caused COVID-19. And if you see here in this picture, you have a big virus here, which is showing in green the spike proteins, the membrane protein here. This is the envelope protein, and you have this circles in the nucleocapsid inside. And it's quite a big virus in terms of having almost 16 non-structural proteins. And when it started sometime in January, when we found out in 2020 in China, majority of the laboratories, majority of the groups started working on this particular protein, which is a spike protein, to generate a vaccine, considering that this protein has a major role in terms of the interaction of this virus with the human cells. And how it happens, if you see this pictogram at the bottom, let's say this is the spike protein on the virus surface, and you have the cell which is actually expressing ACE2. ACE2 is the receptor for this particular virus, and it actually expressed in majority of the tissues. And if you see this, what will happen that, in the next step, the spike protein will interact, through the ACE2 receptor, and then it will allow, the further entry of this virus inside the cell. What happens if you can actually block this interaction of spike to its receptor, ACE2, via antibodies? This had been shown an example. Then you actually stop the entry of this virus inside the cells. And if you stop the entry of this virus inside the cells, then you actually reduce a possibility of having a high viral load and the severity and the further persistence of the infection. So this particular theory, this particular concept was utilized almost by all of the groups across the globe to develop the vaccines, which actually majorly targeting spike. I will not say almost all all the vaccines. There are still some which looked into the other possibilities of including many other the proteins of the virus. We'll touch upon it. So something, when we started in our country, sometime in March, we started seeing the cases, and then slowly it goes up. We actually, try to understand how our population actually responding to this virus. And we have shown, sometime in October, we released this information, which is now available in this particular journal in Frontier in Immunology. And we showed that in our population, in a pre-pandemic time, when the CO2 was not there, we already had a lot of CD4 T cells, which were cross-reactive and which were responding very robustly to the SARS coronavirus 2. And probably we acquired them from the common cold viruses. And we also found a second observation, which we provided, that the individual, those who recovered from COVID-19 with a very mild symptoms, actually have very durable cellular memory. This study was done until 6 months, but later we have now seen until 12 months and now 16 months that it is actually, still persisting in terms of particularly the CD4 T cells and the B cells. We didn't see much of the contribution of the CD8 T cells. So this was the first observation that we quickly made in 2020, and that becomes the basis for us to actually look into many other aspects of this infection, and also the vaccines which were actually rolled out in the country. So when we talk about vaccines, before we see the vaccines in our country, there were many platforms which were actually touched upon across the globe. Majority of these platforms which either utilize the only RBD protein or the spike protein as a subunit vaccine. There were platforms, those who utilized just by inactivating the virus, utilizing some chemicals. There were platforms, those who looked into the live attenuated vaccines, as well as new platforms came in because of the need and because of the necessity at that time in the real time of controlling the pandemic. Something which were came up in a two major ways were the adenovirus based vaccines. The adenovirus based vaccines, which were replication incompetent vectors, where they were actually capable of having the spike protein of the virus, and the DNA or the RNA vaccines. The RNA vaccines, which were, I think, largely utilized and has been largely studied as well across the globe, particularly the mRNA vaccines developed by Pfizer and the Moderna. So these majority of the platforms were actually thought upon because of the existing knowledge on the vaccines development, as well as the understanding on the immune response to SARS-CoV-2, as well as a lot of knowledge from the SARS-CoV-1 and MERS, which we have seen in 2003 and 2011. And what happened is that, of course, there was a lot of preclinical development, and the clinical development happens. And, majority of the target was on the protein subunit, as well as on the recombinant viral vector vaccines, and very minimal on the virus-like particle or the live attenuated vaccines. But slowly that changed with a lot of input from basically the protein subunit vaccines with the combination of different adjuvants. And what we saw here in our country, in 21, that majority of the vaccine that rolled out were actually targeting two major platforms. One was the viral vector platform, which was basically on the adenovirus, the backbone, and the two was the inactivated vaccine, which was actually based on the inactivating the whole virus, not only having the spike protein, and it was developed by Bharat Biotech. We also had some of the approaches and platform developed for the RBD and spike, but we didn't see much progress on that. And now now the scenario is very different. We have this RBD or the spike based vaccines in our country, via of course the collaborations with the outside India, and they are also now in the use. what we will do is that we will just look into these, the vaccines and see what kind of mechanism, what kind of immunological mechanisms these vaccines may actually include. And I'll just, skip this slide just by saying one sentence that, we were always seeing if traditionally the vaccine development for all these kind of platforms, it took like 10 to 15 years, even longer. And for the SARS coronavirus 2, it was reduced to hardly 1 year, 1.5 years. And that was not because we compromised, somewhere in clearing these vaccines or understanding this vaccine. It was because we had developed a very different strategy where these phase 1, phase 2 trials were actually overlapped, and the phase 3 trials were done at the same time. The production was done at the risk, in terms of if phase 3 goes well, we can take up, the whatever we are producing. So by reducing this particular phase, which actually comes down from 5 to 7 years of the trial to months, and where it was very quickly reviewed by the regulators, it actually helped a lot in terms of developing this vaccine in a minimal time window. So now let's see, just giving some example of these vaccines that, what kind of mechanism they might be having. And I will give you, a recent observation from our laboratory on one of the vaccine which was rolled out in our country, Covaxin, that how it might be working and what kind of immune responses it is generating. A very superficial information on that. So let's talk about this mRNA vaccines. Basically, it was initially developed by Pfizer and Moderna. Now we have, one of the, the company in our country, those who are also producing it. It's in, I think, the final stages of the trial. How will it work is you will have the nanoparticles inside which you will have the mRNA, which will be producing spike, and these non-replicating mRNA will be, given intramuscularly, and then you will have these nanoparticles going towards the different cells, and it will be taken up by the cells, where they will release these mRNAs. It will then, inside the cell, you will have the spike protein generated, which will then go up either on the surface as well as in the circulation. And then it will initiate the cycle of vaccine response, as we have seen initially, the overall kinetics starting from innate responses, antigen presenting cells to the lymphoid organs, and the production of antibodies and different cells. It is basically vaccines were actually targeted on the spike protein. They utilized the lipid nanoparticles. They were only used until today by the intramuscular routes, believing and we seen that they have a very strong immunogenicity. Basically, Th1 polarized CD4 T cell response, CD8 are also now seen. Initially, it was not expected to have a very strong CD8 responses, but now we see they also have good CD8 responses. Neutralizing antibodies were nicely made. And initially they were utilized in 2 doses, but now in many of the countries it had reached to even the 4th dose, like in Israel, they are utilizing at the 4th dose. But this was the first time use of these vaccines in human. The other vaccine which comes as a viral vector vaccines, which were, particularly in our country, was, using this AstraZeneca vaccine, which was produced by Serum Institute. And also Russia has produced Sputnik. There are many other vaccines came out later. Johnson & Johnson has provided on the similar strategies of different combination of these adenovirus vectors. How do they work? The scenario is like these non-replicating viral vectors going inside the cells, producing that spike protein. The spike protein against coming up, on these antigen presenting cells, and then, presenting it to the T cells. That whole cycle of T cell, B cell interactions, as well as the killing T cell development takes place, and then you have an overall immune response generated. There were some concerns about these vaccines, particularly, that they may have weak to strong immunogenicity. There's not much understanding available at that time. Now we have a bit of understanding that it induces a lot of, immune responses. Basically, the CD4 cells have a Th1 response. CD8s are also seen, and you have a high magnitude of these antibodies produced by these vaccines. And some of the vaccines, like Johnson & Johnson, were only utilized at 1 dose, but now, like for other vaccines, it has been utilized as 2 doses. There's still, the concern was there previously, the concern is still there, that what will happen in terms of the pre-existing anti-vector immunity as it is generated by the adenovirus. Once you have the immunity against that virus, will that allow the further doses of this vaccine to really induce or perturb the immune responses, or to boost up the immune responses? The vaccine subunits, which is, which were just a small part of the virus, either the spike protein or the small RBD, domain, which is a receptor binding domain, which actually interacts with the ACE2. They were actually utilized in these vaccines, and some of these vaccines are now already in use in our country. And how do they work is, you will have these small proteins, which are, let's say, mixed with an adjuvant. they will taken up by these antigen presenting cells. They will induce the maturation of the antigen presenting cells because of the adjuvant, and then these antigen presenting cells will initiate that whole cycle of adaptive responses in terms of producing the antibodies as well as the T cell and the B cell memory. We believe, in terms of the previous and the historical observations, that, the immunogenicity may not be as strong as we see with case of the adenovirus or the mRNA vaccines. And the CD4 T cell, the polarizations toward Th1 and Th2 is basically depends on the kind of adjuvant they are using. The CD8 may be weak, and we may need multiple doses. We may need multiple boosters to really reach to a level where, you can have a optimal, sufficient quantity of protective responses and to have the memory development. Something which was, also utilized was the COVID vaccines, which were actually inactivated viruses, where the viruses were actually inactivated by utilizing the chemicals. And I think we have seen this is a very traditional method. And one of this vaccine, which was, largely talked, was developed by Bharat Biotech, which uses a TLR7/8 agonist adsorbed to alum. And this adjuvant has been used first time in human, and it becomes something which is of high interest for the immunologist as well, to understand how this is actually helping in terms of inducing the immune responses or the immune memory. There were many other countries, including China, those who produced it. The mechanism of providing immune response may be something which may not be very different as what we see in case of the subunit vaccines, but it will actually include many other proteins of the virus that may have a kind of beneficial repercussions on the time of exposure to the virus, particularly in the current scenario. I'll just touch upon that at the end of the slide, that they will actually be received by these antigen presenting cells, and they will just present it on their surfaces, and then they will induce a cascade of T cell responses, B cell responses, and the antibodies. And again, like the subunit vaccine, it depends on the adjuvant, that what kind of polarization they will have. Remind that a lot of Th2 response may not be very good in terms of the future exposure of the virus, that may actually mediate towards some kind of pathophysiology. So mostly the focus of these all vaccine developers is on to provide a Th1 polarized vaccines. It was thought that CD8 responses may be weak because, of course, it is on the basis of previous observations with many of the inactivated vaccines. Here, the multiple doses may be required, and in terms of responses, it may have responses to the non-essential epitopes, but it may also have the responses to many of those epitopes which are actually necessary. And one point that I just would like to mention here, that these inactivated vaccines may have a superiority over the spike-only vaccines, because the spike is highly vulnerable for the mutations, right? So if you have a lot of mutations, you are actually affected and impacted by these variants, those who are having a mutated spike. But the other proteins, other than spike proteins in this particular vaccine, may actually provide a supportive, a kind of complementing help, a kind of protection, which do not affect by these variants, because they do not have a large mutations in other proteins of the virus. And a coordinated response is actually necessary for the protection. So that's where these kind of vaccines, these kind of platforms may actually give us much more superiority in terms of our fight against this pandemic. Just to give you, a little bit more overview on, what level of understanding we have on this vaccine, is this particular vaccine, which is an inactivated vaccine, as of high interest in our countries, was actually developed by Bharat Biotech. It was actually developed utilizing this, D614G variant, and the virus was actually isolated from a traveler, and it was mixed with this adjuvant, which is the TLR7/8 agonist. It's adsorbed to alum. Initially, it was given as a 2-dose intramuscular injection. It was given 4 weeks apart. When the trial happened, when this vaccine was trialed for phase 1, phase 2, and later phase 3, many of the questions they actually answered in terms of whether it induces antibodies, whether it is efficacious in conferring protection. Yes, there was, of course, different studies came up with the different factors of from 90%, 80% to 70%, even 60% protection in different populations in later in the real world data. But many of these questions which were unanswered during the trial were whether this vaccine is capable of inducing humoral or the cellular immune memory. It was not very clear how long that immune memory may persist, and whether those memory responses can actually respond to the recently circulating variants, and that becomes a very important questions at that time when we actually have rolled out these vaccines. And I think there was a lot of hope on this vaccine which was developed in India. So we addressed some of these questions. I will not get into the details. What I can just give you an idea that what we did, we enrolled the individual, those who received this vaccine, and we compared their immune responses in terms of antibodies, CD4 T cells, CD8 T cells, and the antigen-specific T cells, which are targeting different proteins of the virus from spike to RBD to the nucleoprotein. And we also try to explore that what level of immune responses this vaccine is generating. Is it reactive? Is it responding to the different variants, like the variant that come from UK, from Brazil, from South Africa, and from from India, which we found Delta. And we compared these responses to find out the extent of immune response this vaccine is generating with the mild disease recoverers, right? The individuals, those who recovered from the mild COVID-19. So this study was actually taken up in collaboration with multiple centers, and we actually detailed it out in terms of what kind of immune memory it provides. And it is now recently accepted in Nature Microbiology, and it will be soon available in the public domain. I'll just give you the brief information that what this vaccine is doing is, if you see this particular slide, just go one by one. If we see the RBD binding antibodies, this vaccine induces antibodies that binds to RBD, but in terms of their impact by the variants from UK variant to South Africa variant and the Delta variant, these RBD binding antibodies are largely affected. So these variants impact a lot of these RBD binding antibodies, giving us the message that RBD alone is highly vulnerable by their influence of any variations in these variants, the mutations in the spike. When we look into the neutralizing antibodies, we found that against UK it is fine, it is not much impacted, but again the South Africa and Delta is actually influencing it. In terms of memory B cells, they were not very different what we see with the neutralizing antibodies. There was an impact of these variants on the memory B cells induced by this vaccine. But more interestingly and impressively, what we found that the CD4 T cells, which are induced by this vaccine in memory almost until 6 months, they are not hampered, they are not affected by the mutations. So it's believed that these CD4 T cells might be helping a lot when we are exposing to the virus in the second time after the vaccination, because they are still very helpful in tackling these variants. So not the antibodies, but the T cells might be actually leading at that particular stage when we are seeing the mutants in our country. And we found something interesting with the CD8 T cell, which was a bit expected, that a very lower, the magnitude or the response to the variants was seen. So with this particular work, we actually have shown that this inactivated vaccine has the capability of inducing the immune memory, and the immune memory can actually resist or can actually restrict the variants in terms of causing the disease. Something which I will just end up the talk before I take you to the key takeaways is to control, while we see many of these vaccines, some of the vaccines were also studied outside India, which were like particularly the mRNA vaccines and the subunit based vaccines. A lot of their immune responses were studied. And what we found is that almost all of them has the capability to induce the immune responses. Some of them may stay for some time, some of them may stay for a longer duration, for 6 months or 8 months, but they are boostable. You can just boost them because they actually induce memory. Something which has not been done by these vaccines is all vaccines were given intramuscularly. They all gave you the response, the memory systematically. But the virus, if you see here, what happens in natural infection, you actually get the virus via intranasal, via nasal route, right? And because of this, you actually develop a lot of memory responses and a lot of protective responses in these areas, upper respiratory tract or in the lungs. With this systemic vaccination, we may have a time duration after which the systemic the immune responses can reach to these areas, particularly in lungs, and then help to reduce the severity of the disease. That is what we have seen, but that will not protect you from infection. And I think that was well seen by almost all these recent scenarios where we talked about a lot of breakthrough infections. Yes, infection happened, but not the severity, because you need time from systemic immune response to reach to these areas and to control the virus. So something now what we need to really control this pandemic, and not only to control this pandemic, but also to see in future how these respiratory pathogens can be actually tackled, is the intranasal vaccination. so that we can actually induce the immune responses in these areas of upper respiratory tract or the lower respiratory tract, which is the site of entry. And immediately after the entry, they can be tackled by the immune responses, and you will not generate infection. If you don't allow the virus to replicate and multiply, you do not transmit that, right? So you want to protect the infection and transmission, and for that you actually need the vaccines which are working via intranasal route. So with this, I will just end up here with the key takeaways. The key takeaways for our audience here is the most frequent vaccine failure is the failure to have been immunized. Please take vaccines. And protection may persist after the disappearance of vaccine antibodies, right? The vaccine antibodies may go down, but that's where the immune memory comes into picture. This particular vaccine that we studied, a Covaxin, which is an indigenous vaccine, induced immune memory, and it consists of neutralizing antibodies and the CD4 T cell responses. And it also has a larger breadth of T cell memory against the variants. And I believe that the booster immunization with the intranasal vaccine is now the way forward to prevent the infection and the transmission. And I think the most important part is here, where the future is to actually harness the understanding of the immune responses to these vaccines, so that we can design much more efficacious, much more potent vaccines for the kind of pan-corona or the future pathogens. So these are the faces here, those who helped us in this journey, past almost 5.5 years. Co-passengers, we can say, those who were with us for some time. Some of the PhD students were there. They all are doing, the great in there in the area in the virus immunology, and they helped us. They helped me particularly initially to establish this particular lab with a lot of human immunology programs. And it was not possible, without this network, which we were able to generate from, the multiple clinical and academic centers in India, as well as the academic centers outside India, from USA to Oxford, UK, as well as in France and Japan. And of course, you can understand the human immunology and these studies, are not very, they need a lot of funds. And it was only possible because of this support that we received from our different agencies, both at the national as well as the international level. So we are really, appreciating their help and their faith in our programs, that we can really, bring some, the difference in terms of talking about translational immunology and talking about vaccines in the country, and bringing up some information for the vaccine design. And with this, I just stop here, and I just thanks so much to you for your patience and for your attention. And I'm ready to take up any of your queries regarding this particular topic. Thanks so much.
Thank you, Dr. Gupta. It was really a fascinating understanding of how the whole the field has developed, and how how we are how where we started and where we are here today. There are plenty of questions that I have, and there are a few questions that have come up. The first question that I would like to ask is something else, which is, when the COVID-19 sort of broke out and in the early 2020, were you, given that you are an expert in the field, were you really confident at the time that a vaccine could be found within, say, 12 to 15 months? Was it even with all your experience, having seen the kind of time it had taken in the past, what were you thinking at that point in time?
Yeah, I think that's a very interesting question. Something which is, we were also very, kind of a scenario where we cannot predict at that time what's going to happen. But, we had a lot of confidence that the knowledge that we had developed on SARS coronavirus 1 in 2003 and MERS in 2012, that was actually there. A lot of vaccine trial had happened, but they stopped at somewhere like phase 2 or something, because there was not much interest of the developers or the agencies to go further. But there was a knowledge. So when these, particularly the groups came in, particularly, from US and also from India, they were talking about utilizing a traditional platform. We had a lot of hope that yes, maybe in next, 8 to 10 months, we will have something in hand. But we were not expecting that 8 to 10 months, we will have it in human. And that was something which was really amazing to see, that, by that duration, we were actually able to take it out. Yeah.
I mean, for me, as a, lay person with some understanding of science, what I can only think of it as a miracle. From the time it hit, from the time we could see thousands of people dying, and a total, fear that had spread across the world. To the time the news about the vaccine coming up, to the actual rollout of the vaccines and the scale, what the world has done. I mean, I'm not talking about this country, that country. What we have seen in the world, I think for your field, this is like a sort of,
it was amazing.
It's a dream.
You see that even I put in that slide and which showed them how even the vaccine developers actually came forward and did those overlap studies, took a lot of risk at the financial level. The government supported a lot that, okay, let's take a risk and let's do these overlapping things. And regulators, the things that they take 3 years to evaluate a vaccine, they actually finish it in a month. And I think those things helped everyone to move together.
Absolutely. Because what we were seeing at the other end in terms of number of people dying, we were ready to, accept. I mean, the emergency clearances were given. People took the vaccine, though there were always, the naysayers, people who don't believe in, the anti-vaxxers, etc.
Yeah, that will always be there. Yeah.
So the large majority of people accepted this, right? And that I think it's a fantastic endorsement for the entire scientific process, and that's one of the heartening things from my side. Okay.
Yeah.
Now, looking at the very the future, I mean, in a sense, what we are really talking about is that a major episode happened, a certain reaction happened, and we are now we feel far safer today than 2019-20, right? Now, there are some, there are questions like, for example, V.K. Srinivasan asks, we have done whatever it is that we have done, but can we think of a one-time single shot vaccine for COVID-19? Is anybody even looking at, for example, like polio, right? Polio vaccine, you do it once and you are safe. Now, is it even possible for COVID-19, or are we, is there some understanding that we need to have as laypersons around this?
Yeah, I think, there is always a possibility for a single shot and, having a long-term protection. And if something which is something which we still have to develop a lot of knowledge, right? And that's what, as I said, my team is looking at those areas. If we really find out, what you can induce that can lead to 40, 50 years of protection, then you can take it out while you are developing a vaccine. Okay, you should have checklist 1, 2, 3. If you have it, it may lead to the, the long-term protection. So it's possible. Only thing is that we have to first generate a lot of understanding about what is happening in a scenario where you are seeing a best persisting or durable protection, and then come back and design rationally that particular vaccine.
Okay. I know there's a question from Rachana Acharya, which I think still a lot of people probably have in our country and around the world, is will it be useful to take the booster of COVID vaccine? Is it even necessary? Now, give us a scientific perception and answer.
Yeah. So, why do we need a booster? We need a booster to, let's say an example. I have took 2 doses. I made 20 T cells. I made 30 antibodies. Just a very layman term I'm trying to explain. Now they remain at that level, right? What the booster will do, it will actually induce the number. It will increase the number, but that will not be the only one outcome of the booster. The second outcome will be the memory cells that we have actually made on the way. The good quality memory cells will persist for some duration, let's say 6 months and 9 duration, not a very bad quality. So the high affinity clones, I will say high affinity cells, will stay in circulation. Now, when you give a third dose, what you are doing, you are actually increasing the number of good quality immune memory. And that is necessary not to give you protection against the ancestral virus, is necessary to give you protection against the variants, because now you will have a broad protection. And that's where a third dose is actually good to enhance our immune system to have a broad protection against the future variants.
Okay. There is a question. So I hope, Rachana, you heard that, and all the others who are still thinking, just go and take the boosters.
Yes.
Yeah. So here's a question from Vishnu Narayanan. One of the several questions that he has asked is, even in herd immunity, people with immunity will still act as a carrier of the pathogen, right? I don't get how they actually reduce the spread.
Yeah, yeah. So in case of the herd immunity, if we talk theoretically, I will not give the example of COVID-19 here. I have told this that these vaccines which you are making right now, what we are using, they are systemic vaccines. Why they are allowing the transmission of virus? Because they are not having the immune memory at the site of entry, right? If I now talk theoretically, if I have a vaccine which is actually working at the site of entry, you will not have the infection. If you don't have the infection, you don't have sufficient virus that can transmit to a next person. And that's where the herd immunity works. So don't get confused with this recent scenario of talking about COVID vaccines and the herd immunity. It's different.
Okay. So I'm not I'm going to ask a question which may sound a little political, but that's not my intention. When different types of vaccines came up, mRNA, the name sort of, it was a, for us again laypersons, that was a new name. Whereas the adeno virus back ones, particularly the Oxford University SIA one that came to us, then the Covaxin, whole virus, or dead virus vaccine. I may not be using the right term, so I apologize for that. So you have these different versions that came up. We saw in the popular press, like, this is better than that, that is better than this kind of a thing, which never really, at least the popular reading really did not go into details, or it might have talked about the details, but it was more at a very superficial level. So I want to learn from you, these multiple paths people took for their own obvious, solid scientific reasons. Now, from the expert point of view, with your understanding of all of this, right? Why were these different choices made by different people? Was it because it could be done quickly, or there could be many reasons, I don't know them. So each of these companies and even the countries took certain directions. Can you tell us a little bit about why, and the efficacy rates of each one of it, a little bit, without getting too much into the politics of it? Is that?
Yeah, I think a very straightforward answer to this: why different manufacturers, developers, or different groups look into different approaches. One of the possibilities at the time of pandemic is what you are already working upon, the
Mhm.
approach, the platform that you have. You have a lot of basic data about that platform, and if you have that, it actually reduce again the time which you want to get into the the vaccine development. And that's where the groups like AstraZeneca at Oxford, they were actually already working on these adenovirus vector platform vaccine from past 2 decades. So they had a lot of knowledge on it, so they just moved ahead with it, right? Because at that duration, at that juncture of pandemic, it was not necessary to only go with the subunit vaccination. We didn't know what's going to work because we didn't know how we are
Right.
responding to the virus. So that's the answer to the one question, that why different manufacturers, developers took different approaches. The second question that you asked about the efficacy, the head-to-head comparison of the different platforms. Recently, now 2 3 vaccines has been compared, but it's a bit difficult because you are not doing that in the same individual, right? You are talking about the different cohorts, different population, different individuals from different demographies, and then comparing the data. That may give you an understanding on what kind of immune response these different vaccines are generating. But when it comes to efficacy, as we already discussed, these are systemic vaccines, and they will definitely help you to control the disease severity, whether it is an adenovirus vaccine, whether it is an inactivated virus vaccine. So in that way, I think we should not much worry about what is best. Best is something which is not giving you a disease severity, which is protecting you to reach to the hospital. And that's the best at this scenario where you just don't have anything in your hand. So, and the third thing about the different platforms, how they are working, and what kind of their immunological, the inputs or the outputs will be. Definitely, they will all have a very different way of inducing the immune memory or the protective immunity. Some of them may be, a kind of comparative in terms of the T cells or the B cells. Some of them may not, like antibodies. They may have a different titers. But I think we should not much worry about what is the quantity coming out of these different vaccines. We should worry about what is the quality coming out from these vaccines. Let it be less quantity, but if it is a good quality, we are fine. We just want to protect from the disease.
Yeah, that's a very good clarity that I can get out of that. There is another question from Vishnu Narayanan, which is, if we generate memory for the immunity, will it become a trait through the evolution?
Yeah, I think that is something which is a very interesting question. And whether getting exposure to different viruses with different pathogenicity, our population, our the society is slowly evolving to fight against multiple, needs to be explored. I think that is something which really need to be explored in future in terms of the different pathogenicity. And I think it needs next 1-2 decades of the programs which we really run, and it's really important because I think the frequency of pandemic will not be the same as we have seen in past. I think the frequency will increase now as we are very close to the nature. We are getting into those areas where these viruses, the new viruses are there. So I think we should learn about this particular process, how the different progenies that are coming actually now in future are helping or are fighting against the different pathogens, the level of resistance they have.
Or can we see it a different way? That in history, there is this process that has already been going on, in the sense that there have been all kinds of crazy viral forms have been coming up, and the human capability to resist them has also been developing. And every once in a while, one major breakout happens, like the Spanish flu that we read about, and maybe some more in the past, which we haven't had documentation, and may have been restricted and limited to plague we have always heard about. Now, after many decades, we have been hit with COVID-19. There were few, the general SARS, MERS, Ebola, all kinds of threatening ones had come out, but nothing hit us the way COVID-19 hit us. So the next one, again, there could be all kinds of, so this is a breakout purely from a statistical point of view. I'm not talking about the the biological capability, etc. If I were sitting at a distance and I'm merely looking at episodes of crazy things happening, this is an outlier in terms of the way it has actually hit us. So probably another such outlier hitting us is not immediate, because it's like does lightning strike twice the same place within a, the probability will be just too tiny, right? Can we see it that way? I mean, I'm not saying we should take our eye off and rejoice.
That's an interesting viewpoint, but I can just tell you, being a virologist, I can tell you
Right.
one thing. We were hit by this virus by almost each and every traditional, a kind of principle of virology. Almost each and every step it hit us. It actually it said that, we said, oh, this will affected by a lot of heat in the country. Come on, nothing happened. Heat has actually even increases the so I think the future, it's difficult to predict, but what the viewpoint you have, it's interesting, and I believe that, what you said should be there. At least we should not be, coming again to this pandemic so frequently. Let's see, what the future has for us. Yeah.
Again, it's like, in a sense, COVID-19 or this particular virus with all its variant as a one single large group, again, this is not designer one. As various talks happened, it just so happened that it had all the weapons that it could hit, it could spread, it could just kill a person. It could do just about everything. If you're talking about the hero in the viral space, this was it. It could do everything that nobody else could do.
Yeah.
And to really again make a similar combination happen may not be that easy. But that doesn't mean that your job is easy. You're still looking out for crazy ones. Now, one thing that has happened actually, again from for us, we keep hearing all kinds of crazy news, monkeypox, or every once in a while, every month or so, we suddenly see a new virus has been found out or observed in China. Invariably, China's name keeps coming up, or elsewhere, and because we are always comparing that with again COVID-19. How should we as laypersons even look at such news?
Yeah, I think in this, during this pandemic, there was a lot of misinformation, came out, and still, like for monkeypox and all these diseases. I think I will suggest that link to the real authentic portals, either from the ministry, from India, or like from WHO or from the other ministries, which are health ministries of the other countries. Don't go by this a lot of knowledge from WhatsApp or the Facebook, which is actually really detrimental. So, yeah.
I'm really happy that because of an event like this, we have institutions like you who are normally behind the scenes are coming out, and a lot of information is particularly with the social media, with Twitter, with all the bad things that are happening in the social media. You also have the good things where there are authentic handles, there are web pages where up to the minute accurate information is being presented from the health ministry, from at state level, at central government level. So that's a good thing. So we know at least a place where we can go to and get this information.
Yeah.
And hopefully, it can be spread to people instead of the false news. Now, back to the questions. So there is another question from VK Srinivasan, where he's talking about there was a news that the use of different type of COVID-19 vaccines for dose 1 and dose 2 give more effectiveness. Is that true? So by mixing and matching, so the first dose is one vaccine, the second is another vaccine. Is there any study, any understanding that you have that you can?
I think particularly from the countries like UK and now from states, and also from from Germany, we have this information where this heterologous boosters, we call it as that, initially someone who has received adenovirus vaccine now received the mRNA. They say that the efficacy and the effectiveness, particularly of the immune responses, is a way higher. So it seems like the heterologous settings are actually working much more efficiently. If we talk about our country, I think until today we do not have much of the information, but I can just tell you informally that some of the studies are going on in our laboratory as well, where we are looking into these multiple combinations of the vaccines which were rolled out in the country and their heterologous effect. And I think in a few months we will be able to bring that out in collaborative terms. Yeah.
So normally when we are talking about any pandemic, right? So there is one is the immunological approach. The second is trying to come up with medicines, right? So after a person is affected, how do we treat that person, kind of a thing. Somehow we could see that in this particular period, the immunology response was much faster, stronger, and is actually saving us. But we have not actually found the treatment side of things, or antiviral medicines, the early stage, late stage. So we don't have a reliable answer. Every protocol that was proposed by our own medical fraternity, they have sort of withdrawn. We could see during the horrible Delta wave, all kinds of medicines talked about, all of which we have now gotten away because that's not really efficacious. Can you see any again? You may not be directly working on it, but you would know from the other research areas. So what is happening in that sense? Can you tell us?
I can being a virologist, I can tell you one thing. We in when I was in Pasteur, we used to love the viruses because of their smartness, right? They are really clever in terms of the way they cause the infection. We have to understand one thing here. For any therapeutic modality, what you want to control the virus. Do you think an individual who has not developed the infection, who has not developed the clinical manifestation, will go to the hospital for the treatment? No, they won't go, right?
Right.
The time they develop the infection and the pathogenesis and the manifestations, then they go to the hospital. This is the time when you already have a lot of virus which has caused the pathology. So at that stage where if you give such treatments at a very early stage when the viral load is very low, they may show some effectiveness, and that has been seen in some of the settings. But majority of the clinical trials failed because they reached to that area where you have hospitalized individuals where virus probably has already done its part. And now what is happening is the virus-induced immune pathology, where the virus may need to be controlled, may need to be reduced, But then now you have to control a lot of pathology which has already initiated, where the antiviral may not help. So having an antiviral is really tricky in case of the infections like this, where the virus replicate very quickly, very fast. So that's what probably more the scientific reasons why we did not see a lot of efficacy of these antivirals develop. Maybe we have to find something which could be utilized in a very early stage if one knows that they have a virus. And that's what, if you have seen, I will not go into the scientific detail there, but that's what we have seen in many of these drugs were prescribed by the clinicians during that Delta wave. Someone who tested them, because of the close contact, they did not develop the symptom, but they started their medicines, they did not go to any kind of severity. That is a lot of anecdotal information available, and that all falls into that area. The duration, the time when you are actually doing the treatment, that's more important.
Okay. The two other things that we have now we hear, one that happened where we heard a lot about it during the Delta wave is the cytokine storm. So that a lot of people died because of not the virus, but what the immunal system in the body was causing. That's one. The second, I want you to answer both of this together. Now, a lot of people are talking about the long COVID, the impact that the person is going through, having lived through COVID, treated, came out, but they just can't come back to normalcy. There's a large number of people.
Yeah.
Can you tell us a little bit more about what is the scientific community thinking about doing, studying in trying to address these issues?
Yeah, I think, this is not very specific to COVID. This has been seen even with previous virus infections. And if you see historically, a very, very close example I will give you is chikungunya, right? In chikungunya, when we see in 2007-8 in India, we did not see a much of the, severity or the, the mortality, but you might have seen still the guys were affected neurologically or developed arthritis or many other consequences which were pathological, and they're still suffering from it. So in case of viruses, what will happen? It actually induces a lot of immune responses, and that some of these immune responses may attack the self. And that's where we may have some kind of, autoimmune or the immune pathology that generates. The groups, many of the groups in our country are now actually looking into it, that what kind of these low COVID, manifestations are, and what are their relations in terms of the immune pathology. And I think not only in India, even outside India, there are many groups those who are studying it, because it becomes a problem once the pandemic goes down. I think long COVID will come into a very, a kind of prominent problem for us. So I think in some time we will have some of the answer. I will not say one will be able to really crack that scenario, but we may have some answers why it has happened and how we can really, control it.
Okay. So the couple of other questions before we end the session. This is now.
Badri, let me ask a very fundamental question, because I think we might want an answer on that.
Sure.
Is the pandemic over? And do you make a call, or is it a political call?
No, so I think whether the pandemic is over or not, it depends on what kind of infection is still spreading. We are still seeing there are infections. We are still seeing there are individuals infected with the virus. It has slowed down at some of the places, which we are now believing it may turn into an endemicity, where it will come once or twice in a year at a particular place, not everywhere. But it's all in a kind of illusion, right? We do not have a real evidence which can say that this pandemic is now over. I think we still take some more time to really, come to that point to say and make a statement. Yes, the pandemic is over. It may enter into endemic scenario.
So let me ask so the answer is no, it's not over. And second, I think you're saying it's a very regional thing. So from region to region, you're going to have different responses. And so it will be determined regionally. So you may have local pandemic rather than a local epidemic rather than a national, global pandemic.
Exactly. You may see much more spread, much more infectivity at a particular area, and that's what we call scientifically endemicity. And I think that's what is going to happen. But right now, pandemic is not over. We still have to be very careful.
Okay. So the question that naturally comes is that until now, if you take a student's career, what they were really looking at, getting into, etc., would have had a major impact over the last couple of years in terms of what a student wants to study is triggered by the heroes in the society, right? They look at a pilot and then they say, hey, he's flying a plane and I want to be a pilot, right? Now we have seen a new set of heroes emerge in the space. What is the educational track one needs to take to get into this particular space that you have been working on? And what opportunities are there in the Indian context?
Yeah, I think like particularly in my lab, we try to influence the students starting from their 11th and 12th class. My hypothesis and strategy is, if I can feed 1-2 words into their mind about the immunology and about real impact of immunology onto our society, then they will start thinking about it, right? That is something which is largely lacking in our system. The educational system do not allow to expand the possibility one can actually have in their career. And that's where I think the pandemic in terms of these areas has impacted positively, that many of the the youngsters now, they are coming, like many of the emails and the messages I am getting, that they want to really get trained into this area, and they want to know how they can actually develop into this area. So getting into it, you can go from being a clinician, you can get a doctorate into these areas, get yourself much more depth knowledge, and then go further for the further postdoctorals. You can even go from your masters, where you can learn about the different microbiology, biochemistry, virology backgrounds, go into do the much more rigorous doctoral programs, and then get into the further these things are really lacking. What many of the students I found, they feel it becomes a curriculum, right? You do BSc, you do MSc, you do PhD, and then what? No, that's not good. We should understand why we are doing bachelor's and master's and why we are doing PhD. A PhD is a necessity to get a particular expertise in one very narrow area. And a postdoctoral is further a necessity where you can get more expertise on it. So I think those things needs to be much more spreaded around the the student community, that how should they decide their career and their future. They have to understand at a very early level. And that is not just 1 or 2 traditional fields. They can actually contribute a lot. And I think youngsters in India, they really are amazing. They really are amazing. If they get a good direction, I think we will do wonders.
So then correspondingly, all the, or not all, at least major colleges and university across the country will have to have specialized programs, build labs, provide training programs, maybe build up an internship relationship with national labs. So this whole body, this whole infrastructure at the educational level has to be now created, right? Which your traditional regular programs had not really bothered as much. I mean, I'm sure there were some colleges, some universities that always had programs, but we just now realize that we have to have it multifold. I mean, if it was there earlier in 2 colleges, now we need it in 100 colleges, right? So is there any national policy program that is underway to encourage this kind of a thing?
Yeah, I think there are a lot of discussions, and a lot of these policies are now probably going to come, where we will have a lot of emphasis on developing these either the virology and the immunology domains to have much more prominence in the country. But still, we have a lot of opportunity already existing. What we do not know, that these the opportunities lie here, and the person who needs that opportunity lies here. There is a gap in between.
Huge information gap.
Yes.
Yeah,
that's what is required to fill, and to really make aware the students at a very early stage that what is possible, where it is possible. And I think that will make the difference itself.
Gopu, do you have any other questions or announcements? You are on mute.
Yeah, I think we've had a lot of questions, unless our audience has any questions you want.
No, I've covered all the important questions anyway. Dr. Gupta, it was a pleasure hosting you and interact with you. Not just during the lecture and afterwards the question and answer session. And I could clarify a lot of my own questions. Thank you. Thanks a lot for coming and then presenting this lecture today. On behalf of Arahamira Science Forum, we thank you for a wonderful lecture. Thank you. Yes,
thank you.
Our next month's lecture is actually also on medicine. Dr. Valiyathan of Kerala will give us a lecture on Charaka Samhita, one of the most ancient treatises on medicine. So I want to make the announcement.
Sure. Thank you. Thank you. And by all, do you want to say anything, Dr. Gupta?
No, I'm fine. Thanks so much, and I really appreciate the efforts that your forum is making. And I will really be more happy if you can spread the awareness in terms of not only the knowledge, the expertise areas, but also how these youngsters can really get into those lines. Thanks so much. Thanks so much for the invitation. It was a real pleasure for me as well to interact with you.
Thank you. Thank you.